Is my voice is audible? Hello. Is my screen is visible? Now? Is it audible, sir?
The way your voice is buffering, it seems. Can you?
Is it now okay? Is it now okay, sir?
It is audible, but there is some noise in your voice. Maybe you can try without your mic or earphones.
Now I'm audible clearly. Sir, is my voice is audible now?
Your voice is audible, but it is not clear. Maybe try changing your mic. That might work.
Okay, but screen is visible, right, sir?
Yeah, screen is visible.
Sir, now my voice is clear.
No, there's some disturbance in your voice, it seems. That is the issue.
Sir, now my voice is audible.
Oh, Divya, I guess the issue is same.
Sorry, sir. What?
Oh, Divya, the issue is same. Your voice is buffering in between. There's some noise there.
Okay. Sir, is my voice is audible now?
Yeah, now it is clear.
Okay, sir. Okay. I'll share my screen now.
Yeah, sure.
Screen is visible, sir?
Yes, screen is visible and you are audible perfectly.
Okay, sir. Thank you. Good afternoon, everyone. So, participant has reached the count. So, we will start our presentation. Myself, Divyadashini, PhD scholar, Department of Food Process Engineering, National Institute of Technology. So, herewith, I'm going to give the practical demonstration and the result interpretations of my knowledge on the topic of nutritional composition of the food compounds. So, I have listed out some experiments so we can deal with our experiment also demonstration. I have clearly mentioned the video presentations, and with that I will be also solving your question and answer session at the end of the lecture. So with that we will move ahead.
Good afternoon, everyone.
Yeah, I have started.
Okay, then
So
we'll start. Okay.
and with the table of contents, these are the things I have want to discuss with you guys today. So starting with introduction, origin and significance of what is proximate analysis, what is macronutrient composition, and comparative study with between the methods. Usually I have clarified most common methods we have studied, then going with the experimental procedure, then video demonstration and results and discussions, and key outcomes. I have created a summary table also, and question and answer session. And so introduction is about what is proximate analysis. So this is an effective quantitative method which is analyzing a different macronutrient which is available in the food component. So for example, if you are taking a cookies with a various amount of moisture will be there, and crude protein will be there, and crude fiber. So crude means the term, it is not a classified fiber or a protein, so it is in the term of extracted crude. So that's why I mentioned crude protein and crude fiber, crude fat, and total ash. So if you are taking fiber rich biscuits, for example NutriChoice, we are getting more amount of crude fiber comparative to the Mom's Magic. Since I'm not specifying on the brands, but I'm saying an example. So protein, as well as for example we can classify the protein based upon their dairy products and all, and nitrogen free extract. So if we are going with approximate analysis, we will go for digestible carbohydrates, like which is including all the nitrogen extracts also, irrespective of the carbohydrates samples present. And so origin, so what is proximate analysis I have discussed. So origin is, it was first developed in 19th century by the scientists Hennenberg and Stockmann in 1860. So at the experiment station in the Germany. So initially it was a standardized method for evaluating the composition of animal feed. So we know that animal feed like silages, fooders and all. So for that they have started an experiment. So how much they are, like for humans also we need an like nutrition dietetic charts are there. So for animals also, they have started a standardized method in order to evaluate the basic composition which is available. Then the same analytical framework was followed for our food analysis also. Starting with our lab, accredited lab, they started fulfilling their water nutrition labeling requirement. And so they started with this proximate analysis composition. So it is a basic fundamental procedure which will be understanding of nutritional composition using simple laboratory techniques. And then here I have seen that nutritional evaluation. So first significance importance is nutritional evaluation. Obviously we will know how much nutrient we are taking, not only the food samples and the tasty and appearances and all. We have to know that how much nutrient intake we have going in a per day, in terms of calorific value or in terms of per servings also. So usually in the labeling and all, they will be keeping as a per serving. So if you get like in a layman term, it will be in a servings. So if we want to express them in a percentage like that, we can also use it in a percentage manner. Then quality control used by the food in the processing industries for estimating the raw material qualities. And next one is shelf life assessment. So this is indicator of the food stability, like moisture and fat samples, we know that. So if the sample is high in moisture and it is open to the atmosphere, so oxygen attraction will be there obviously. So it will be developing a mold growth, and it will lead to the raw material spoilage also, even if it is a full product without the packaging or any other external protection, it will lead to the damage. So for that we need to estimate what type of packaging we want to produce. So for that also we need estimating the moisture content. So for in that case this analysis will be helpful. Then economic importance. So for example, if you are developing a product or a processing methodology, so we need to analyze what are the instruments or what are the technology or what are the raw materials we have to incorporate for developing the product. So these are the economic importance will play an important role here, because cost estimation value assessment is very very important. So for example, if there is one pomegranate juice, and same pomegranate juice was fortified with probiotics or any other vitamins or minerals, so the rate obviously will change. So the terms called it is fortified with probiotics, fortified with vitamin A and C, like that. So for this also it will be essential. Then research applications for our peoples. So if you are going for the RSM or design expert methodologies, so first I will be utilizing the normal parameters, starting with the protein content, moisture estimation. So if I'm developing a product called a protein powder from some waste resources, so I will be starting with the packaging shelf life and sensory evaluations and the processing. Then I will format from that optimized product, and then I will be developing a product. So like that it will be useful for research applications. Next, so these are the list of experiments I have planned: moisture, fat, protein, fiber, ash, and anthocyanin and chlorophyll content. So this is a basic layout. First, I'm talking about the food samples, then how we will be going with the sample preparation. Then I will show the instrument handling and data analysis also. So this will be the step by step. So starting with the moisture content determination. So moisture content is known that the free moisture as well as the bound moisture is there. So here I'm terming as complete, both profile. So moisture refers to the amount of water which is present in the food samples. Here the moisture content is determined by drying the sample and measuring the weight loss. So obviously it is the weight loss at a predetermined temperature and predetermined weight in the predetermined duration. So the after the all the moisture evaporation, there will be the residue, which is called as dry matter content. So we can also calculate the dry matter content once the moisture content is determined. So there will be two methods, common method available. One is oven dry method, another one is moisture meter. So in oven dry method, so I will explain the features first. So there will be two common methods are available. One is oven dry method, another one is moisture meter. So oven dry method, here the principle is similar. So water will be removed by heating the sample at constant temperature, which is 105 degrees Celsius in a hot air oven. And whereas in a moisture meter, we will be measuring the electrical infrared properties using the halogen heated lamp. We will be heating the sample in the powder. Then type. So this is a gravimetric, which is gravimetric means it is a weight based. Here it is instrumental, where it is a rapid determination of moisture. Then accuracy, which is very high, and in AOAC method also they are using this moisture using the oven dry method only. And the moderate accuracy will be there, which is affected by the sample type and calibration. If the instrument is not calibrated well, or if you are not putting the sample before tearing that instrument, so there will be some issues with the sample accuracy. Then time required, so it will take 3 to 6 hours of time depending upon the food type, and also in it will take few seconds to few minutes. So for example, time required means if it is high moisture food, so it will take 6 hours of time. So if it is low moisture, for example dry leaves, like that, it will take 2 to 3 hours. Then sample preparation, this is requiring simple steps. For example, in a drying oven, and we need a weighing balance and desiccator with a silica gel. So silica gel is a moisture absorbent. It doesn't allow the moisture to enter, and it will be absorbing any excess moisture which is present in the atmosphere. Then suitable for the laboratory, and both are suitable for laboratory analysis. If in case of moisture meter, so we will be using the industrial applications and quick checks. Then limitations are there. Obviously it is time consuming, where it is very reliable and heterogeneous, and also you can use for high fat foods. Then output will be, there will be a formula derivation for oven dry method, whereas in moisture meter we can go directly with our analysis, which will give the moisture percentage directly. So coming to the importance, so it will be helpful in determining the shelf life and storage stability, and affects the texture, taste, and microbial growth if the moisture is not correctly predicted, and it will be leading to the inappropriate storage conditions with the packaging materials also, and critical for nutrition concentration and processing behavior. This is very essential for the composition behavior also. And we will see the videos once I will show how it is. So this is a hot air oven. So once I have taken the sample, which is correct, then I have to reduce the size of the sample. So it has to be in a uniform thickness. So that's what the drying behavior will be good. Then this is a container, and we can use a cuvette or a petri dish for our benefit. And here hot air oven, which we have to keep our sample inside, and once we getting the sample, we will be placing in the desiccator to cool down. So here we are taking three weights. One is weight of the dish, and W1 is weight of the dish plus sample. So if I am taking the 5 grams of sample, I have to put 5 plus the weight of the petri dish, which might be 31 to 28 grams, like that. And W2 will be weight of the dish plus sample after drying. So we can't calculate the sample once it is hot. We have to keep in a desiccator. Once it is cooled down, then we have to take the weight. So with this also we can calculate the dry matter percentage. So 100 minus the moisture percentage, after getting from the calculated value, we will getting the results. In moisture meter, I have a video demonstration for moisture meter also. We have to keep the reduced sample size, because if we are keeping more sample, it will take more time to determine, and measuring the sample size with the accurate weightage, then analyzing for 5 to 6 minutes. Once the final moisture content can be obtained.
Now we are seeing the video demonstration for the Penser moisture meter, and we can see the indications, and we have to place the sample in the moisture plate, and we have to evenly distribute the sample for the efficient moisture transfer. And after that we will be pressing the start, and the moisture meter will be on. And after that of 2 to 3 minutes of time, we will be getting the direct moisture meter reading. Here I have taken the powder sample, so the moisture comes around 2%. And after that, since the sample is destructive, we have to discard the sample and we should not use further. And we can see the calculation part for the moisture determination by the oven dry method. So here we will estimating the moisture percentage, which is W is weight of the dish which is taken, and W1 is weight of the dish plus sample, and W2 is the weight of the dish plus sample after drying. So usually we will be taking 5 grams of samples. So here I have taken the carrot sample, so I am putting the values and I am getting the results as, so the moisture content of the carrot sample is 45.45%. We can also give the triplicates and duplicate trials also.
So this graph plotting is for the oven dry method. So I have also listed the how we have to put the graph plotting for the moisture content versus time.
These are the results for the oven dry method. Here we can see the hours of drying as well as the sample moisture weight. So we have taken 6 hours of drying and control sample with other 5 samples. So we can also yield the graph moisture versus time. So for that we have to select the data and go to insert, and we can use 3D column or 2D bar. Here I have taken the line chart, and then after that we can see the legends for all the samples, and also we can see the moisture percentage at the verge of the points. And like this way we can also calculate the differences between how the control sample and other samples are reacting. And for if I click this point, we can also see at which point the bend or the curvature comes.
So that's it for the moisture determination. Moving to the next one, which is a crude fat estimation. So fat estimation will be based on the extraction of the fat using the non-polar solvents, which is where the non-polar solvents means the electrons are equally sharing. There will be no charge, partial charging. So that polar solvent we are using, for example, petroleum ether or hexane, and the fat will be dissolving in the solvent and it is separating by the evaporation, where there will be three methods in the Soxhlet apparatus. I will explain later, and leaving behind the extracted fat residue. So that fat residue we will be collecting, and that will be termed as crude fat percentage. So most common methods are Soxhlet extraction and continuous solvent extraction. Both are similar, and Mogineer or acid hydrolysis for the dairy or the liquid samples. So first, Soxhlet extraction I will be explaining. In the Mogineer or acid hydrolysis, for example, we will be in Mogineer, we will be using the ammonia solution and then ethanol for the extraction. Then acid hydrolysis, we will be using HCl for hydrolysis purpose. And then when the fat layer is separated, we will be skimming off from the dairy product or high fat products. So I'm going with explaining the Soxhlet apparatus now. So first, processing of a sample on the apparatus, there will be three parts. One will be thimble, where we'll be holding the sample. We have to place in the filter paper. We have to put inside the thimble, and after that it has to be attached to the flask. Either it is called as a fat flask or extraction flask, and we have to attach the flask to the condenser where the thimble is attached to that. So placing a thimble inside the Soxhlet apparatus, extraction will take 6 to 8 hours. So it has to complete the cycle, 6 to 8 hours of cycle. And then once the cycle is over, we have to evaporate the solvent and weigh the extracted fat. Here I can show you. So this, when we are getting, this is the called as thimble, placing the sample inside the thimble. So then we will be solvent filling in the inside the thimble, and we will be attaching the thimble to the Soxhlet apparatus. So in collecting the extracted fat. So calculation is very simple. So W, we will be calculating three weight as it is from the moisture also. So W1 is fat of weight of the fat and extracted sample. So it is at the end we will be collecting. Then W2 is weight of the empty flask before weighing. So for all the experiment, we will be weighing the empty flask before putting the sample or before the extraction proceeds. So make a note down on this. So weight of the empty flask or empty dish or empty cuvette is very, very important. Then only we can know how much sample we have taken. Then weight of the sample in gram. So ether extract on a dry matter basis also calculated. For example, so if I am not keeping the sample in the oven for 10 to 12 hours before the extraction, so I can go with the extraction on a dry matter basis. Once we are getting that, there will be formula crude fat obtained. For example, if I'm getting 12 to 13%, so 12 into dry matter percentage, for example, I'm getting 10%. So 12 into 10 divided by 100, that will be your ether extract on a dry matter basis. So this is the thing. Then so I have said the solvent for fat extraction is non-polar. So what is the best solvent for our particular food samples? In the first, I will explain the food samples with the type of fat and suitable solvent. So cereals and pulses, there will be mostly neutral lipids. There will be there, either it is in the fat form or in a lipid form. There will only be complex lipids. So it will be extracted easily. So we can go with the petroleum ether, which is a non-polar lipids, will dissolve well easily. Then dairy product, which is high in fat. So fat, which is also with the phospholipids and carbohydrate. Lipid is attached to the carbohydrate tail. So in that case, we can go with the diethyl ether or a chloroform and methanol mixture. We will have to go with the polar solvents, or either in a single solvent, or we can go with a double, which is a mixture of both solvents. For example, chloroform and methanol in the ratio. Then extracting both the neutral as well as the bound lipids. So neutral lipids will be extracted easily. When comparative to the bound lipids, it has to take some efficient extractions, and also we need some proper solvent to extract them. Then meat and fish, this is also containing complex lipids, like it is bounded to the protein. There it is bounded to the phospholipids tail. Here it is bounded to the protein. So chloroform and methanol 2:1 ratio we can go, which will breaking the protein lipid bonds. Then baked foods like neutral fats and emulsified oils are there. Here in this case, we can go with petroleum hexane or ether, which is for efficient for the free fat extracts. So there will be three considerations for choosing the right solvent. One is based on the fat type as well as solubility. Another one is based on the extraction method. Another one is boiling point, reactivity and toxicity, and cost and availability. So for example, first one is based on the fat type and solubility. So if I'm going with some biscuits or cookies, so we can go with the non-polar solvents because it might be consisting neutral fats and oils or triglycerides only. So polar solvents we can go for either fat, for example paneer extraction, like that. So polar solvent like diethyl ether or chloroform, which is in the mixture also we can be going, are used for when the phospholipids, glycolipids or bound lipids are present. Then based on the extraction method, so Soxhlet apparatus will be there, acid hydrolysis method, which I have mentioned for the liquid sample. So we can use based upon their safe and economical use and easy evaporation also we need to understand one another consideration. Then coming to the boiling point, some solvents are for example ether, hexane are having common boiling point, so it should be evaporated easily at the range. Then it has to be non-reactivity and non low toxicity. So some sample might be tend to react with our solvent, so we have to make sure it has does not react with the sample, and low toxicity should be there. So you can go with the organic solvents. Then cost and availability. Based upon if I am going for fat extraction, if my entire project is depending upon fat extraction, you can go with the availability and the cost efficient solvent also. And in summary, so if the food sample contain many free oils or fat, we can use a petroleum ether or hexane, which is called as common solvents. If the lipids are in a complex or a bound form, we have to use either in a polar solvent or in mixed solvents like a 2:1 ratio of chloroform or methanol. So I will show the video of Soxhlet apparatus now.
This is the video demonstration of crude fat estimation. Starting with sample preparation, we have to finely ground the sample for efficient extraction, starting with instrumental setup and folding the sample in filter paper and using the solvent. Here I am using petroleum ether, pouring 90 ml of petroleum ether.
This is a lab scale thimble. So this is a small thimble where we have to keep inside our sample. This is a sample I have kept. We have to fold that using a filter paper. We can take 2 to 3 grams of sample. And if we are taking a big amount of sample, we need to pour more amount. So I have taken 2 grams. So I'm pouring 80 to 90 ml of solvent, which I have taken petroleum ether.
In the flask. After that, we will be loading the sample and locking the flask and opening the condenser valve for the efficient cycle and programming the temperature and let this solvent starts to boil. Once it starts to boil, we can note down the condenser timing. After 6 hours of the fat extraction, we have to close the valve like this and let the sample be recovered from the solvent. We have to evaporate.
So I'm saying the solvent should be evaporated from the sample. For that, we can also do the solvent recovery method. We have to run another one cycle. Otherwise, we can go with the rotary evaporator also, which is available. And then here, for example, if the sample as well as the solvent is mixed, we have to keep that in a water oven for 40 to 30 degrees Celsius for 10 minutes so that all the solvents will be evaporated. So we can get only the crude fat remained in the flask.
The excess solvent associated with the fat extracted and moving into the solvent recovery, we can use this solvent for the same fat sample analysis. This is the extracted fat sample from the Soxhlet apparatus. Okay, now we are moving into the calculation part. It is very simple that we are subtracting the amount of the fat from the sample together which is available in the flask and divided by the amount of the sample we have taken into 100. And also we can get the ether extract which is on the dry matter basis. Once we get to know the dry matter percentage using the moisture content analysis, we will automatically get the ether extract on the dry matter basis. So I have taken those two samples to show the differences. So first one I have taken the butter cookies and another one I have taken the leaves which is less in fat obviously. And after the calculation I am getting the fat yield of 27.8 percentage and the leaves for 5.1 percentage. So this is the way we will be calculating the fat yielding percentage.
So that's it for the fat estimation using the Soxhlet apparatus. Next,
This is the
moving to the crude fiber estimation. So crude fiber is nothing. It is just an indigestible portion of plant material such as cellulose, lignin content, hemicellulose content. So it is represented the structural carbohydrate. We can term the name as structural carbohydrate in the food. So principle is simple. We will be digesting with acid digestion, then following with the alkali digestion. So for that, I will let me explain first reagent, then I will explain the principle. So we need defatted sample. So we can also get the sample what we have did in the Soxhlet also, that is a defatted sample that inside the thimble. We can recover that from the thimble and we can use that for the crude fiber estimation if the sample is similar. Otherwise, we have to again go with the defatting. So we can use a continuous salt oxalate extraction for that. And then defatted sample, then reagent is 1.25 percentage of sulfuric acid, H2SO4, then 1.25 percentage of NaOH, which is sodium hydroxide. So these are the reagents required for the crude fiber estimation using acid alkali method. So first one is we will be processing that with a dilute acid for 30 minutes. So that will be simulation for our gastric juices which is available in our intestine. So that is a similar type of process we are doing here. Then going with the alkali digestion. So the remaining residue after the digestions will be there called as the crude fiber. So it is dried, weighed and incinerated. If some carbon materials are there, it can be incinerated for the ash content. So here in this image we can see that there is a defatted sample. And going with the acid alkali digestion, you can use a hot plate also for this. And filtration using a filter paper, I have used Wattman filter paper 41 for this. And drying the residue, I have taken the leaf samples to show how much fiber content. So leaf will be good example for our crude fiber. Then ignition, and we are getting the ash residue. So moving to the procedure part. So first we will be taking the defatting sample using a solvent, as I said earlier. Then weighing 2 grams. We can also go 5 grams, but I'm taking 2 grams for my experiment. So weighing defatted sample and 2 gram and keeping in a 500 ml flask. Adding 200 ml of 1.25 percentage of H2SO4, boil gently for 30 minutes. We have to boil them for 30 minutes. Then filter and wash with the hot water, 3 parts. 3 parts in the sense, if I'm using 200 ml, I have to use 600 ml of hot water to wash it. In like it is like 1:3 ratio. So until it is acid free, then going with that, we have to skim off the residue from that filter paper without tearing the filter paper. And we can also use a muslin cloth. It will be useful, like if it will be good. And transferring the residue to the clean flask again, adding 200 ml of 1.25 sodium hydroxide solution, and again it is boiling for 30 minutes. After 30 minutes, filter and wash with the hot water, then dry at 105 degrees Celsius to maintain the constant weight. So it might be with the solvent, it might be with the water. So we need to get the constant weight before going to the incineration. Incineration means burning in the muffle furnace. Ignite the dry residue at 550 degrees Celsius in a muffle furnace for 3 to 4 hours, cooling the desiccator and weigh. That weigh is called as W2. So there will be two weight only. One weight is the dried residue which we kept in a hotter oven, and another weight is the ash content weight which we kept in the muffle furnace. So W1, which is a dried residue minus weight of the ash after the ignition divided by weight of the sample, which is 2 gram into 100, so that we can get the crude fiber percentage. So this is the important analysis under the nutrition labeling. So indicating a dietary fiber content in the food. If the person is having a bowel syndrome or some other issues with their stomach, they can't digest a high fiber food. So for that, we can also go with this analysis and indicating dietary fiber content in the food, helping analyzing a digestibility and quality of feed as well as the food material, and high crude fiber indicating a low digestibility, obviously. So high crude fiber is indicating low digestibility. So we can optimize the raw material for any product development, which is important for feed formulation also. Then this is a video demonstration for crude fiber estimation.
Now we are seeing the video demonstration of crude fiber estimation. Starting with reagent preparation, we have to prepare 1.25% of H2SO4 and 1.5% of NaOH solution, and we have to take D5 sample for this analysis. First, we will be going for the acid digestion, pouring 200 ml of this acid.
So in this video, I have taken the defatter sample. This sample which I have taken for the cookies in the soft set apparatus, that only. So I have taken 2 grams of this powder cookies.
For the acid digestion, pouring 200 ml of this acid and boiling for 30 minutes in the hot plate. So after that, filter and collect the wet residue using the Wattman filter paper. Later, the wet residue has to be washed with the hot water in order to remove the acid adhered to it. It has to be 6 parts. So if you are using 200 ml, you have to wash it with 600 ml of hot water. Then going with the NaOH solution, we have to wash it and filter it, and after that this has to be done for the alkali digestion. Later, drying the residue and keeping for the ash in the muffle furnace for 6 hours. Later, that ash has to be collected and used for the calculation of crude fiber. Now moving to the calculation part, we are calculating the crude fiber percentage using the weight of the sample initial value and the weight of the dried residue before the ash ignition and after the weight of the ash after keeping in muffle furnace. So W1 we are getting and W2 we are getting after the ash ignition. Then divided by the initial amount of the sample. I have taken 2 grams of sample and calculating the crude fiber percentage using the formula. So the leaf sample contains 21% of crude fiber.
So that's it for the crude fiber calculation. We will be moving to the another experiment called crude protein estimation. So there are various methods of crude protein estimation. For laboratory scale, I'm going with the Lowry's assay. So this is most commonly used calorimetric assays. So it is discovered by biochemist Oliver H. Lowry, hence the name of the protein estimation called Lowry's method, which is developing the reagent at 1940s. So this is a image which is 3 major steps. One is reagent and sample preparation. Another one is reaction with the sample, and then measuring the absorbance. That's it. And calculating the standard curve using a graph. So principle is, first one is the peptide bonds which is available in our sample will form a complex that will be reacting with the copper ions under the alkaline condition. That is the first reaction. Second reaction is FC reagent. We are introducing the FC reagent to that sample, so that copper peptide bond complex in the first reaction will subsequently change the color into the blue. So that blue color we will be observing in UV spectrometer. So that will be indicating our crude protein content. So these are the advantages why I have taken this is, so there are different methods is also there, like a bioweight test and all. So it is sensitive assay which requires no protein digestion. In Zeldol method, there is a 3 steps like digestion, distillation, and titration. Here we will be going with only the reaction with sample and UV absorbance. So it is 10 to 20 times more sensitive comparing to the ultraviolet absorption at 280 nanometer. Then more reaction, more specificity will be less affected by the turbidity of the sample. If the sample is more turbid, we can also control our turbidity and we can estimate our protein using a UV absorbance. Then it is more sensitive than an anhydride reaction, but the pyruvate reaction is less sensitive than the pyruvate reaction. So it is simple to perform and it can be used in a small scale labs. So disadvantages is color will be developed, will be differing from the protein to protein. Even if you are taking the same sample with a different concentration, so the color will be differ. So the less constant than the pyruvate reaction and more constant than the absorbance, which is in the anhydride reaction. The color is not exactly proportional to the concentration. So which is the main step for our thing. So first one is solution A. So only two solution we are preparing. Solution A is 2% of sodium carbonate in 0.1 normality of sodium hydroxide. So 0.1 normality is mostly used in many experiments like acidity, titratable acidity and all. So normality we will be calculating using C1 V1 is equal to C2 V2. So C1 is a concentration of the stock standard and V1 is a concentration a volume of the stock standard is equal to C2 is a concentration which is 0.1 normality into what volume we want. So V2 is a volume of our required sample. So if we want a 10 ml, we can put 10 ml in the place, or we can put 100 ml also. So I want 100 ml, for example. So I'm taking 0.4 grams in 100 ml of distilled water. So which is getting the 0.1 normality of sodium hydroxide. Then 2 grams. Once it is completely diluted, then we will put in 2 grams of sodium carbonate to the 100 ml of solution, stock solution. Then solution B, which is copper sulfate 0.5 percentage and 1 percentage of potassium sodium tartarate in weight by volume. So we will be keeping in the mind if it is a weight by volume or volume by volume. Then preparing fresh by mixing the stock solution. We have to prepare separate, then mix the both stock solutions together. So after that alkaline copper solution, which we want for the first reaction, which I mentioned in the principle 1. So which is 50 ml of reagent A, which is 2% of sodium carbonate and 0.1 normality of sodium hydroxide, mixing with 1 ml of reagent B, which is copper sulfate and potassium sodium tartarate solution. So that will be our solution C. And solution D is diluted Folin reagent, FC Folin catalytic reagent, which is available commercially. So that, for example, if I want to use 0.5 in the procedure, we will be using 0.5 ml for each test tubes. If I'm taking 10 test tubes, 0.5 into 10, which is 5. So 5 ml of FC reagent and 5 ml of 0.1 normality of HCl. That has to be prepared once we started preparing our standard stock solutions. So experimental procedure is, first, we have to pipette out 0.2, 0.4. Before explaining the procedure, I will explain how to prepare the standard curve. So for this, I'm using a bovine serum albumin, which is stock standard solution preparation. So which is representing 50 mg of BSA in 50 ml of distilled water in a volumetric flask. So there will be two solutions. Don't get confused. There will be two solutions. One is stock standard solution, another is working solution. So I'm explaining now is stock standard solution. So first we will be collecting 50 mg of BSA and putting in a volumetric flask, which is getting 50 ml at the end. So in that 50 ml, we will take 10 ml of this stock standard solution, dilute to the 50 ml of another flask for the working standard. So the diluted sample from the 50 ml of BSA will be calling as working standard solution. So this will be representing 1 ml of the solution contain 200 mg of protein. It is like a standard procedure. So here, we can see the graph. We can see the table where 0.2, 0.4, 0.6, 0.8, 1 ml of sample and diluting them with the 0.8. For example, if I'm taking 0.2 of BSC standard, I have to make to 1 ml, right? So for that, I will be using 0.8 ml of distilled water. So then concentration of the protein and the volume of the reagent C will be using, and then incubating at a room temperature for 10 minutes. Then volume of reagent D, which is 0.5. I have said that 0.5 ml we will be putting, and dark incubation for 30 minutes and absorbance at 660. That's it. So for plotting the graph, once we are getting the absorbance, I will also show the video demonstration. Before that, I will explain a brief about that. So plotting the value of protein concentration on the x-axis and the value of absorbance on the y-axis, constructing the straight line through the points, presenting the values of absorbance. Once we get to know that, we will be getting the graph graph in a straight linear line. In that we will estimate the equation and r square value also. So I will explain the procedure now. Here we will be pipping out 0.2, 0.4, 0.6, 1 ml of working standard. That working standard in the BSA. In a series of the label test tube, don't forget to label, otherwise we will get confused. Then I have taken a 1 ml of the sample for another test tubes also. So making that to 1 ml in all the test tubes. For example, if I am taking 0.2 ml, I have to make to 1 ml, right? So we have to use 0.8 ml of distilled water. Then adding that 5 ml to the reagent C to all the test tubes, including the test tube which is also labeled as blank. So blank is like for UV absorbance, we will be keeping blank. So the blank will be here, our blank is distilled water only. So, and then unknown sample, which is our unknown sample is our test samples. For example, if I'm taking milk, that sample is known as unknown. So we don't know the concentration of protein, that's why it is termed as unknown. Then mixing the contents of all the tubes by either vortexing or shaking the tubes and allowing to stand for 10 minutes. That is called as reaction time, which is we have mentioned in principle 1. Then adding after the 10 minutes, we will be adding 0.5 ml of reagent D, which is diluted with 0.1 normality of NaOH. Then rapidly mixing them well and incubating at the room temperature for in a dark incubation we need to keep because it is light sensitive. So for 30 minutes. Now recording the absorbance at 660 nanometer against the blank. So the plotting the standard curve by taking concentration of the protein on x-axis and absorbances on the y-axis. So I will show you the video of video demonstration of the protein calculation.
We are seeing the video demonstration of crude protein estimation starting with the experimental setup. All the test tubes and the beakers are clean and sterile, and the reagents are prepared, reagent A, B, C, and the reagent D, which is FC reagent using NaOH solution, and I have taken the milk as a sample. Starting with the standard stock preparation, I have taken 50 mg and diluted with 100 ml. From that, I have taken 10 ml and making up to 100 ml. From that, I am using as a standard. So starting with the test tube 1, I have taken 0.2, 0.4, 0.6, 0.8 and 1 ml as a BSA standard. Then to make up to 1 ml, I have been using the distilled water to make up to 1 ml. For example, if I am using 0.2, I have to make up with 0.8 ml of distilled water. Like this, I have to prepare. Then I am going with the sample preparation. I have taken the milk, so the sample has to be 1 ml. So to equating all the standard reagents. Then interaction of this reagent with the sample, starting with a sample reagent C. So reagent C, I have to pour reagent C 5 ml to the solution, and then we have to keep for 10 minutes at the incubation. Then going with the reagent D. Reagent D is 0.5 ml.
So this is the color of the diluted FC reagent with 0.1 normality of NaOH
in each test tubes, including the standards, and after that 30 minutes incubation and moving to the UV absorption at 660 nanometer. As I said earlier, distilled water will be serving as a blank. Okay, after we are
So this is the standard curve calculations for the protein estimation using the absorbance value from the UV absorption spectrophotometry.
getting the absorbance value from the UV spectrophotometer, we are going to plot the standard curve equation using the concentrations on the left side and absorbance on the right side. So we are taking the absorbances as well as the concentration as such 0.2, 0.4, 0.6, 0.8 and 1 ml from the BSA standard solution, and the absorbance at 660 nanometer. These are the corresponding values. For plotting the graph, we are going to copy all the data and go to insert section, and where we can see our recommended charts, click inside and go to all charts. We can see XY scatter plot. Use the second one. This will go for our standard equation. Then click insert. then we need we can see that the pointers are in a linear, but we need to confirm using the trend line equation. So using the plus option, we can go, you can also give the legends for our easy marking and go to the trend line. You can see the linear absorbance, the line came, and also you go scroll down more options, there will be two options, display equation on the chart, click that one, and next display r square value. So both came, we can use this to estimate that our model is valid or not. So see that the points indicating the absorbance, so there will be the 5 absorbance value, all the 5 indicating the absorbance, and here r square value is 0.9, and the equation has come, we have to input our absorbance of the test samples, which is an unknown concentration of protein, right? We have to input in the place of y, and substituting that we will see later, and substituting that we will get our known concentration of protein. Once the standard equation plotting has been done, we have to substitute the sample absorbance and we will getting the results. So I will explain one by one. So standard curve equation using the known concentration of standard solution, that is BAC standard solution, which is having a 50 mg in a 100 stock solution, and giving the equation of y is equal to 0.5535x minus 0.0029, which is having the r square value of 0.99. So first step is we need to measure the sample's absorbance at 660. We already did together with the standard absorbance value. So in the table, I have taken the two samples. So absorbances is,
Sorry.
we are seeing is 1.779 and 0.894. So I have been using the dilution factor. I have used a 10 dilution, so 1:10, and using the substituting in the equation to find the concentration. So I'm substituting in the y the absorbance value, and we are getting the x. So x is the concentration of the unknown protein percentage available in the test samples. So here is a standard curve equation. Using the equation, we will be substituting. So after that, we can also write the concentration in the mg per ml or in a percentage of protein. So percentage of protein is nothing but how much that which is the mg per ml into how much amount of the sample taken for analysis divided by the weight of the sample, which is in mg or a gram. So if I'm taking a 1 ml and diluted sample as 1 ml means, so 1 by 1 into the concentration, which is 32.2. So the protein will be 32.2 percentage. That's it for the protein analysis using the Lowry's method.
Okay. So then moving to the next experiment.
We are seeing,
which is total ash estimation, which is simple using the muffle furnace. So total ash referring to the inorganic residue which is remaining after the complete ignition, complete or oxidation of the organic matter, carbon material which is present in the food. So this will be representing the total mineral content of the sample. So here in the image we can see the sample is taken in the crucible. We have to pre-char. So in some muffle furnace we have to pre-char and keep. So there will be no option for the charring in some muffle furnace, but the muffle furnace which I am showing in video demonstration is already attached to that. So there is no need to pre-char. So for usually the muffle furnace will be having that accessibility, but if it is not there, we have to pre-char. So pre-char the sample means that all the smokes will be out. So the carbonous material will be out. So that's what we are keeping for pre-charring. Then going with ignition. Ignition means oxidation of all the organic matter in the food, then white ash, collecting the white ash. So principle is the sample is incinerated, burned at 550 degrees Celsius in a muffle furnace. So all the organic matter will be destroyed, leaving behind the non-volatile material. So that is termed as ash. So we will be taking a 3 weights. One is dry crucible, W1, adding a known quantity of sample. We can take 2 grams of sample and pre-char the sample on a hot plate to remove the smoke, or electric pre-char is there, and place in a muffle furnace at 550 degrees Celsius for 4 to 6 hours until it is gray or white, the ash appears. So then cooling in the desiccator and weigh W3, we can take that as W3, and repeat the heating and weighing until a constant weight is achieved if it is needed. So calculation is, we have taken three weights, right? Empty crucible weight, weight of the crucible plus sample, and weight of the crucible plus ash. So after putting that in an equation, we will be getting. So what is the importance of why we have to do this crude ashes, which will be indicating the mineral content as well as the purity of the sample. So we can see if that is adulterated, or if it is non-purity of raw material, we can see by the color of the ash. Also we can see if it is more with a carbon material or non-carbonized material, also we can see. So which is also used in detecting the contamination as well as adulterations of many protein powders, not a protein powders, like other powders used for the culinary applications, and use the quality control and nutritional evaluation. So moving to the video demonstration, I will show this is a Germany imported muffle furnace. So there is a pre-chair is already attached inside the equipment. So that accessibility is there. So I will be showing that advanced muffle furnace. Okay.
Now we are seeing the video demonstration for total ash determination, starting with instrumental setup. This is the muffle furnace, and we are keeping the sample of size reduced sample of known weight. I'm taking the chickpea flour. I'm using 2 to 3 grams of sample for this analysis. We have to know the crucible weight as well. After that, we have to ensure the safety valve is locked. I'm going with the program setting. There are two different options are available. One is we can go with a pre-programmed thing, otherwise we can set a new program. For our experiment, we are going
In this instrument, usually there is an old muffle furnace is also there. So in that we have to keep that analog temperature only. There is no automation, so it will be heated gradually. So in this German imported one, here we will be getting that the temperature, and it will be stayed at for some time, then it will be gradually increasing. I will also explain this in the video.
With the new program thing, it has been done in a 2 steps. First, I'm using a 250°C for 2 hours, and it will be maintained for 550° for 8 hours, and pressing the N button to save the program. Once we have to check the program setting. After that, all the program setting, we will press start. Then once the program has been started, we can see the graph of a temperature rise. Using the safety valve inside, we can see the status of how our sample is going inside. And after 8 hours of sample ash, we are unloading the sample, and we can see the ash in the crucible. Moving to the calculation part, here we are estimating the total ash percentage using the weight of the empty crucible and weight of the crucible including the sample, and weight of the crucible plus ash after the muffle furnace ignition. So after substituting the values which is available on the right side table, we are getting the total ash percentage of 4. So total, here I have kept chickpea flour. So total ash content of the chickpea flour is 4%.
So that's it for the ash estimation. Moving to the next experiment, which is carbohydrate. So there are various methods of estimating. I'm explaining the total difference method only. So there is different methods in the right side table. We can see there are different methods are there. First one is anthrome. Another one is phenol sulfuric acid method. Then DNS method, dinitrile salicylic acid method, and Felling's Bentic test, and enzymatic methods. So there are types of carbohydrate estimated. In anthrome method, we estimate the total carbohydrate. In phenol sulfuric method, we will be estimating the total sugars. Same as for DNS and Felling Bentic test, we will be estimating the reducing sugar. And specific sugar is also estimated using the enzymatic method. So I will explain all the principles, but I will be explaining the total difference method and nitrogen free extract separately. So principle here is dehydrations by the H2SO4. So acidification and dehydrolysis will be there, and that sulfuryl derivative will be having a color. So that will be reacting with anthron and yielding green blue. So in anthron estimation, we have to be more careful with the anthron. We have to keep the ice bakers or some other cooling environment while handling the anthron. And it will be absorbed at 660. Then phenol sulfuric method, total sugars will be there, and for that we will be same, the furfural derivative will be there, and it is reacting with the, here it is reacting with the phenol, so and yielding the yellow orange color, which is 490 nm nanometer. We will be getting the wavelength. Then dinitrile salicylic acid, there will be reduction in the agent with the amino acids of the carbs and protein, and once it is reduced, so that the yielding the color of orange red, which is 540 nanometer we can estimate. Then Felling's Bendix test, reducing sugar, reduction of copper or cuprous oxide, and giving the red precipitate. This is a visual estimation, so once it is red precipitate comes, we can estimate that. And enzymatic method, specialized enzymes are there, and the color will be vary depending upon the assay. So coming to the main part, so the primary source of energy for living organism is carbohydrate. So carbohydrates is composed of the various organic molecules like carbon, hydrogen, and oxygen. So there are major types which is including the monosaccharides, glucose, fructose, disaccharides, sucrose and lactose, polysaccharides, starch, celluloses, and glycogens. So what is the need for estimating this? So nutritional value will be depending upon our what we are taking. The carbs matters, right? So everyone is suggesting for the overweight, reduce the carbs, because it is a storing forms of energy in the presence of the glycogen, ATP, right? So it is important in quality control and dietetics, also on food formulation and dietary studies. Then used to analyze the effect of processing on a carbohydrate content and helping to estimate the energy contribution from the carbohydrates in the kilocalories. So it is yielding 4 kilocalories, right? So fat will be yielding 9 kilocalories. Carbohydrates will be yielding 4 kilocalories, and protein is also yielding 4 kilocalories. So this is like estimating the energy composition from the carbohydrates once we know how much carbohydrates is present in a per servings or per grams of sample. Then used for dietary planning on food composition analysis, essential in biochemical and industrial applications. Then so total carbohydrate estimation by difference method. One, because we are going for proximate analysis, so this proximate analysis comes under with the difference method only. So carbohydrates is here, it is not measured directly, so it is a difference method. So difference in the sense, so all the major components, some of the all the major components, for example protein, moisture, fat, fiber, everything, and subtracting them with 100. So here the equation, total carbohydrate percentage is 100 minus moisture, protein, fat, crude fiber, and ash. So significance will be providing a quick estimate of total carbohydrates which is available in the food. So useful when there is no direct carbohydrate estimation is not feasible, we can go with this method, commonly used in proximate composition analysis for nutritional labeling. So then limitations is also there. It will be including all the nitrogen free organic matters. There is no variability between the sugar, starch, pectins, or organic acid. So it is yielding all the total carbohydrate content of our sample. And error will be there because it is obviously it is like subtracting. So once it is mistake in moisture or protein, there will be the detection in the accuracy of our results. Then does not provide any information on carbohydrate type or digestibility. So then coming with the nitrogen free extract, another one which is an yielding calories, kilocalories, we can get it as energy content of the sample. So we will be seeing in a nutrition labeling like the energy content of the samples or energy content of the food or the biscuit per serving. So for this they are using this method only. So it is including more digestible carbohydrate like all the digestible carbohydrate like starch, cellulose, hemicellulose, which whatever is available, and in the minor quantities is only. So the total digestible nutrition in kg is crude protein plus crude fiber plus nitrogen free extracts, which is carbohydrate, plus ether extract into 2.25. So 2.25 is a factor which will be multiplying. So I said the kilocalories, right? So carbohydrate is yielding 4 kilocalories, and protein will be yielding 4 kilocalories. But in the fat, it is 9 kilocalories. So 4 into 2.25 is 9. So that's what they are multiplying into 2.25. So the total digestible nitrogen free extract will be reflecting total available energy from all the nutrients in a kg format. Okay. So significance is digestible energy rich fractions of our sample we can estimate. So if I eat this sample, I will be getting this amount of energy. So for that, this analysis will be used. Then used in the animal nutrition to formulate the balanced ratios and comparing the energy content across the samples. Then moving to the anthocyanin determination. So anthocyanin determination, these are the natural plant pigments which is giving from the various fruits like blueberries, uh, cabbages, that the purple color cabbages, which is also rich in anthocyanins, raspberries, grapes, vegetables, flowers, red, blue, and purple color flowers. So these are the compounds is available in our environment which is yielding a good anthocyanin content. Other than that, many products are there, but for easy approach, I'm using this example, this as an examples. So this is belong to the class of organic molecules called as flavonoids. Since it is a broad topic, micros, so micronutrients, so I'm choosing the pigment compositions. So molecules called as flavonoids, this is a group of polyphenol compound which is found in the plants. So anthocyanin, there will be two method estimation. One is pH differential method, another one is UV. Both are UV method, but the difference is in the formula. So I will explain this. So principle is anthocyanin, the light of the, it will be observing the light of the selected wavelength and transmitting the light of the selected wavelength. So that's why they are having the pigmented color. So the pigment is largely depending upon the pH as well as a C ring, which is having a positive energy of the anthocyanidin molecule. So that molecule have the C ring. Because of that positive charge in the C ring, we are getting the molecule as pigmented at the pH 1. So even after the pH 4.5, so for pH 1, we can use KCl or HCl. For 4.5, we can use sodium acetate buffer for our pH maintaining. Then that once the pH is going beyond 4.5 or higher, even that, so the C ring is hydrated, those are positive charges neutralized. So the anthocyanin will be losing their pigmentation and become colorless. So after this pH, the anthocyanin become colorless. Note this one. And anthocyanin will strongly absorb the wavelength between 460 to 550 nanometers. So it will be in the color of cyan blue, blue-green light. So reagent is simple. Alcoholic HCl, we need to prepare HCl. So we will be preparing 1.5 normality of HCl. So using C1, V1 is equal to C2, V2. So 12 is a normality. So putting 12 into V2 is equal to 1.5 into, if we want 100 ml like that. So it will be yielding 12.2, 12.5 ml of HCl for 100 ml. From that, we will be using a diluted ethanol. Mostly ethanol will be absolute ethanol only, it will be in 99%, otherwise it will be in diluted form. Carefully check before doing the experiment. And in this image we can see the grapes, and using the extraction with the using a mortar and pestle, then UV centrifugation if there is any pellet or any the, if we are taking a clear liquid, there is no need for centrifugations. Then absorbance is at the UV. So procedure. So anthocyanin, I have taken the leaf samples or any other samples we can take, and alcoholic HCl we have to extract with that. So that alcoholic HCl is 85% of this ethanol, which is diluted ethanol, 95%. Then 15% of this 1.5 normality of HCl. For example, if I'm taking 8 ml, sorry, 10 ml of extract, so 8.5 ml of ethanol, then 1.5 ml of 1.5 normality HCl. Got it? So after this extraction, filter or centrifuge it and transfer to the clear solution to a new container, dilute to a desired reactions. So we can also dilute that if it is having a higher yield, and we can multiply the dilution factor in the formula and read the absorbance at 535 nanometers and calculate the anthocyanin concentration as per the formula. So here is the formula: absorbance into 535 nanometer into volume of extraction. So into the volume of extraction and into 100 divided by the weight of the sample in grams. So if I'm taking 1 grams of sample, just mention 1 gram into 98.2. This is a standard factor calculation. So results can be expressed either as a mg per gram or mg per 100 gram. So significance is it is a nutritional and antioxidant potential of the food. It is very, very important because antioxidants, it is scavenging the free radicals. So if that formulated food is having a high fat, if the antioxidant is there, it will increase the shelf life. So this is the basic formula for the functional food development. So and then serving as a marker for freshness, ripeness, quality in fruits and vegetables. So once the aging occurs, the leaf samples or any other food samples will be yielding the color change. So this can be determined using this. And there are many AI sensor schemes, but this is a food analytical methods. So then reflecting the color stability, visual appeal in the food products, help accessing the impact of processing and storage on pigment retentions, supports a functional food development, I said earlier, and health claim. So validation, if we are fortifying with any other antioxidants like vitamins or something, so we can also use the health claim validation. So this is acting as a biochemical marker in breeding and crop improvement studies. So I will be showing the video demonstration for anthocyanin determination.
The video demonstration of anthocyanin estimation, starting with the reagent preparation. So we need two reagents. So alcoholic HCl, which consisting of 85% of ethanol. I am preparing 85% of ethanol now. After that, I am preparing 15% of 1.5 N HCl. So as I discussed earlier, so we are preparing 15% of 1.5 normal.
One new to this acid handling and all, we should not use the concentrated acid. So we should not use water, should not put the water in a concentrated HCl. So it will be splashing. So we have to put the diluted one to the concentrated one. So in order for the acid handling.
HCl. So I'm mixing both to get 25 ml of extract. So sorry, extract is reagent. So mixture should be 25 ml. So everything should be covered. So using pipette out method, I am mixing the reagents. Next, starting with the sample preparation. So we need to estimate the sample in gram. So I'm measuring them in a gram. So I have taken the fruit juice for our experiment. So after mixing, then we have to take that 1 ml of this clear solution and making up to 10 ml with this reagents. After that, it will be diluted as 10x factor. So these are the blank, and then centrifugation, if it is required, we will go for 6000 rpm for 10 minutes if it is any pellets are available in the extract. Then UV absorbance at 535 nanometer. So this is a photometry, UV spectrophotometry, setting the wavelength 535 nanometer. So using the blank, we have to zero the absorbance. So I will show you. So blank, then we will be zeroing the absorbance. After that, we will be going for our sample.
So in the case of the blank, whatever we have used for the extraction, if we are going for acetone extraction or alcoholic HCl, so we have to use that extract, the solvent to be our blank. So not a distilled water. Then going with the calculation part.
Okay, now moving into the calculation part. So estimation of anthocyanin with the following equation. Once we get the absorbance at 535 nanometer, just substituting it. So absorbance at 535 nanometer, then volume of extraction 25 into 100 and divided by the weight of the sample. So I have measured in gram into 98.2. So I have used 1 grams of sample. So 0.026 is absorbance at 535 nanometer. So we can say that 0.662 mg per gram, or we can express 66.2 mg per 100 grams of sample. So we can say that anthocyanin content in the fruit juice extracted as like 0.662 mg per gram, or else 62.2 mg per 100 grams of a sample. So that's it for the anthocyanin calculation.
So that is for the anthocyanin calculation. Moving to the other experiment.
The video demo
For chlorophyll determination. So here we will also talking about the chlorophyll as well as the carotenoid, which is having a similar wavelength, which will be partly overlap. So chlorophyll is a primary green pigment which is available in plants, which is responsible for capturing the light energy during the photosynthesis and serving as an indicator of plant health, maturity, and quality. So carotenoids, which are synthesized by all the plants and many microorganisms, but not by the animals. So including humans, we have to depending upon the dietary uptakes. So principle is, so the carotenoids and the chlorophyll compounds comes under the group called tetraterpenoids. So this compound will be having a health, for example, like the polyphenols, which I have mentioned earlier. So which is having a potential health benefit. So major dietary sources, which is including a variety of fruits and vegetables, are having a high carotenoid and chlorophyll determination. So carotenoid, if we get in mind, the carrot will come. So if it is a chlorophyll, we can go with curry leaf. So these are the primary food samples which we already know for the layman terms. So this method is measuring the carotenoid and chlorophyll via the basic spectrophotometry method. According to the Beer-Lambert's law, absorbance is directly proportional to the molecular extension coefficient and the path length of the solution traveling and the wavelength traveling the solution, and C is a concentration of the sample. So if the concentration is there, that is directly proportional to the absorbance. So if we are reducing the concentration, the absorbance is also affecting. So make sure that we are using at the correct concentration result yielding also. So absorption spectra of the extract fruits or vegetables will be contained in the chlorophyll and carotenoids. So therefore, it is very difficult to analyze as which wavelength corresponds to which chlorophyll or carotenoid. So for that, in acetone extraction, there will be different types of solvent extractions also there. So I am using acetone for the extraction. So in this acetone extraction, chlorophyll A and B will be expecting two major peaks. So that peaks will be around 650 and 660. So while the carotenoid will be having their peaks at 400 to 480. So that's why we are using that nanometer in the formula, which I will be discussing later. So the reagent sample, it is very simple. We will take a fresh weight of the plant samples. Either we can go with the dry samples weight also, but it won't be suitable for our chlorophyll estimations. So 80% of acetone we can prepare. 80% of acetone means like, for example, if it is volume by volume, we can go with the distilled water to make up that 100 ml. So like that. And in this image, we can see that I have taken the leaves and I have using the mortar at pestle. I will be extracting. Then this is the, if we are going with that centrifugation, we can go. And then later we can go with the UV absorption spectrophotometry. Then same procedure, that instrument is called as mortar and pestle, which will be using for grinding the sample extract. So grinding the extract and total pigment using 80% of acetone. So centrifuge that crude extract at 5000 rpm to discard the pellets. Pellets means the in the centrifuge tube. After the centrifugation, the above liquid, clear liquid, which will be happening above, will be called as supernet. So the below that discarded, so the pellet will be that other residues are called as pellet. So we need only the clear liquid. So after skimming off the clear liquid, we will be discarding the pellet. Then keeping the supernet and discarding the pellet, and absorbance should be 663 nanometer, 645 nanometer, and 480 nanometer, which are giving the major piece at the particular nanometers. Then estimation of chlorophyll content using the formula. There is a formula, total chlorophyll. Using this formula, we will be estimating then carotenoid. So the carotenoid is also having the absorbance. Once we are getting the absorbance of 663 and 645, we will be substituting that in the absorbance together with the 480 and into the volume of extract divided by the 1000 for yielding that results to be in mg per ml into weight. So the mg we can also display the results as mg chlorophyll per carotenoid per gram fresh weight or mg per ml. So significance is chlorophyll and carotenoids will be together be using only different formulas are there. So otherwise it will be like together we can estimate simply. So indicating the plant health, maturity, freshness as a chlorophyll content will be decreases with aging or the stress. Then we can also estimate if the plant is under stress conditions, or also we can estimate if people are working under the plant substitutes for any food product formulations. So we can estimate if the using the chlorophyll about their ages or stressing. then serving as a quality markers in the fruits and vegetables and the green foods, reflecting the nutritional and aesthetic value. Helps to evaluate the processing, storage, or environmental factors depending upon the pigment stability and the green colorations. So carotenoid indetermination. So carotenoids measuring the nutritional value, as carotenoid is a precursor for the vitamin A. So and the potent antioxidant. So provides insights into the color quality, visual appeal, and important for consumer acceptability of fruits and vegetables and processed foods. Helps accessing the impact of storage, processing, and cultivation conditions depending upon the pigment retention, green coloration, and bioactive potentials. So I will be showing the video for chlorophyll and carotenoid determination.
This is the video demonstration of chlorophyll estimation. Starting with the reagent preparation, we will be having a 80% of acetone using and making it with the distilled water. So I have taken 25 ml of this reagent. Then going with the sample preparation, I have taken the curry leaves, which is a fresh and it's not dried. So I have taken 1 gram, and that is mortar and pestle. Using the reagent, I will be slowly mixing and grinding the curry leaves. So we can see that using the mortar and pestle and the extracts flowing, and I will be mixing in a clockwise as well as the anti-clockwise direction for the efficient extraction. Once it is done, we will be transferring them into the amber centrifuge. Since it is light sensitive,
So this is called amber centrifuge, which does not allow any other light into reflection doesn't happen. So if your sample is having any light sensitive reactions, we can go with the amber centrifuge. So this is amber centrifuge.
it will be taking some reaction time. In the meantime, centrifugation for 15 minutes. I have to keep the timer for 15 minutes and the rotation speed. We have to keep the same sample amount weight for the centrifugation balance. Otherwise, it will make some sound. So then we will be closing the centrifugation panel. Once it is done for 15 minutes, we will note down the timing, and once it is finished, it will indicate with the sound. Closing the centrifugation, then collecting the supernet, and I'm using the acetone as a blank, and we have to zero and wipe that cuvette with the paper for clear vision and UV spectrum transfer. And this is the green extract we can see, and I'm reading the 3 observances using this. And once we get the observance, we'll move into the calculation part. Now, moving to the calculation part, so we are going to estimate the chlorophyll content as well as the carotenoid content. It is like simple formula only. Once we have taken the reading for all the observation, just a substitution from the chlorophyll values, so we will get the carotenoids also. So I have taken the sample curry leaves. So this concentration of chlorophyll A, B, and total chlorophyll, which is the addition of chlorophyll A and B, as well as the carotenoids, were calculated using the following equation. So the equation is as follows: chlorophyll, total chlorophyll as 20.2, at the A represents the absorbance at 645 nanometer. So here 3 absorbance are there: absorbance 645 nanometer, and absorbance 663, and absorbance 480. So these are the 3 absorptions we have taken from the UV spectrophotometer. Then by substituting the absorbance value in the formula, here we can see that the curry leaves I have taken. So the chlorophyll A represents mg. After substituting in the value, it represents mg per gram fresh weight. Since I have taken the fresh curry leaves, it has to be maintained as fresh weight. Even we can also go with the dry weight also. So the carotenoid content as well as the chlorophyll content will vary accordingly. So here it is mg per gram, and total carotenoids here mg per gram fresh white. So we can see that the total carotenoid value as well as the total chlorophyll content by substituting here, absorbance 480 value we have to substitute. After substituting, then into the volume of sample into 1000 divided by the into the fresh weight, which is 1 gram. So after that carotenoid, we are getting the mg per gram fresh white, so 0.028. So results can be expressed as mg chlorophyll or mg carotenoid per gram of fresh white or in mg per ml. So that's it for the calculation part. So this is the estimation of chlorophyll content from the curry leaves. This is the acetone extraction. There are different types of extraction are there. So I have explained the acetone extraction. So that's it for the estimation of chlorophyll content using acetone extraction.
And here we can see in the results itself. So this is curry leaf, so the chlorophyll will be higher. So we can see total chlorophyll will be 0.3 for mg per gram, but the carotenoid is 0.0028. So it is here itself we can see the results in differences. So we can write accordingly. And
This is the
then moving to the summary of our lecture. So I have discussed what I have utilized for our food samples. So moisture content, which is the principle, is the weight loss due to the evaporation of water. Calculating the weight loss at predetermined temperature and predetermined weight. Then key point is measuring the measuring the crucibles or the petri dish, and then cooling the sample. These two are the main consideration here, and maintaining the temperature of the hot air oven, which is 105 degrees Celsius. Then the formula for that same. Then the crude fat, which using a Soxhlet apparatus, either the solvent selection should be clear. We have to use either non-polar or a polar solvents or in the mixture. So if it is a bound lipids, we can go with the mixture of solvents. If it is a non-polars, like the lipids will be extracted easily, we can go with the non-polar solvents like petroleum ether or hexane, and the extraction cycle will be depending upon your fat yielding. So we have to make sure about the timing also. Then crude protein, which is FC reagent forming complex, which is a blue colored complex, which is measured spectrophotometrically. Then here it will be the protein content will vary depending upon the concentration and the dilution factor. So make sure we are multiplying that using the dilution factor formula. Then total ash, the organic matter residue. Once all the volatile matters are evaporated, non-volatile matters residues are calculated using a total ash in the muffle furnace at 550 degrees Celsius for 4 to 6 hours. And we can use, if it is not, if the pre-char is not available, we have to use a pre-char, then we have to keep. Then crude fiber, which is based on the indigestible matter after the acid alkali digestion. So for the acid, we can use H2SO4 1.5 normality. For alkali digestion, we can use 1.25 normality of NaOH. Then dry washing and hot water wash will be there, 3 parts. For example, if you are using 200 ml, we can use 600 ml of hot water for washing at each cycle of digestion. Then this is the formula. So then carbohydrate by the difference method will be estimated using 100 minus sum of all the major components like a protein, fat, fiber, ash, and this is using the data from other analysis also. No direct experiment is required for this. Then chlorophyll, coming to the chlorophyll, I have used the acetone extraction, and then the grinding and extraction using a mortar and pestle, and the centrifugation. Only we need a clear liquid, the supernatant. So if it is having any residue, we have to centrifuge it. Otherwise, no need. We can carry out with the experiment and dilution if it is required. Then the intensity of this green pigment is proportional to the chlorophyll content according to the Beer-Lambert's law. Then the anthocyanin. So the anthocyanins are extracted using the acidified alcohol, which is ethanol plus HCl. So absorbance is measured at 535. I have used the alcoholic HCl extraction. So same will be followed, like measuring the absorbance with the clear solutions. And limitations is for this all the approximate analysis, nutrition analysis. So only the gross composition will be there, no specific nutrition profile is there. So either fatty acids or sugars or amino acids. Then requiring a destructive method, a sample cannot be used further. So if you are using a sample for certain experiment, for example, I said for water which is destructive, after that we cannot be using for any other experiment. We have to use a new sample for that experiment. So if it is in a crude fiber, we have to use a defatted sample. For that case, we can use from the sample from the Soxhlet apparatus also. Then it is time consuming, labor intensive, sensitive to the temperature, light, atmosphere, weighing and handling errors will be there. So even for extracting the standard graphs, we have to make many errors because it can't come in a first time. So that will be there. And carbohydrates are estimated by the difference method, so which can lead to the error propagations also. Then precautions: use an accurate analytical balance and clean glassware should be there. Cool all the dry samples before weighing using a silica gel in the desiccator. So maintain a constant temperature, avoid the air drafts during the drying process, and handle the acids, alkalis, and solvents with the gloves, goggles to protect yourself, and calibrate all the instruments regularly. So whatever we have did for this will be coming under this nutritional labeling also. We can see in the right side total fat, cholesterol, sodium, and total dietary fiber, sugars, protein, vitamin D, which we have discussed in our antioxidant potentials or any vitamin mineral potentials. Then for quality controls, nutrition labeling, product formulation help in standardizing the recipes like baby foods, dairy snacks, and all. And research and development we can use for the supporting new product development. So for example, if I'm developing a high protein nutritional bars, we can go with the high protein raw materials. For that, we need to analyze initially the protein content for the raw materials, like that. And shelf life evaluation tracks the compositional changes during the storage as well as the processing. We can in the morning lecture we have seen the sampling techniques also, so that might be clear for this. And feed and ingredient evaluation used in the feed industries is to balance the animal diets. So this is the conclusion. So it is under the AOAC. Whatever I have discussed is under the AOAC for the global reliability. And this is a fundamental method. It is an initial started in fundamental method. There are many modifications as are there, but this is under the AOAC. So although the traditional will be remaining indispensable, analytical foundation, this is a foundation for the model food analysis. So chlorophyll and anthocyanin content will be reflecting the freshness, quality, antioxidant potentials, and processing, storage, and environmental factors. So these are the helping tools for our estimations further. And these are the references I have taken for the lecture. And any questions, I'm happy to help.
Ma'am, can you again tell what is BSA in the crude protein?
So BSA is a bovine serum albumin. So it is a standard which is using for estimating the crude protein. So it is available commercially. That is like a standard. We can say it is a standard for our unknown concentration. So we don't know what is a concentration. So BSA is a standard for our estimating the unknown concentration sample. BSA is bovine serum albumin.
Okay, ma'am. Thank you.
We can use chats also, like, or in question and answer.
Ma'am,
Yes.
is pre-charring essential for all type of samples before keeping in muffled furnace?
Yes, pre-charring is essential. So whatever I have did in the video is different. So in that, it is a Germany imported muffle furnace. So that muffle furnace is already the pre-char is imported. So pre-char accessibility is there. So it's already starting temperature. It will be already pre-charred. So pre-char is nothing but all the smokes will be removed. If you are keeping any samples inside, so suddenly what will happen? The smoke will be there. So pre-char is important if you are keeping in a conventional muffle furnace. So it will make a error if you are not pre-charring first, and also it will save the times.
And also, do we need to close the sample if we keep it in the lid? Crucible.
No, 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 no. We should not close the sample. So what is the need for keeping? We should not close the sample. We have to openly keep the sample. So I have taken only the open sample, right? So same way we have to keep and take the samples. And we have to be more careful because it is happening at 550 degrees Celsius. Use the appropriate gloves, hand gloves for handling the instrument.
Okay.
Ma'am,
Ma'am.
how to measure the like Mendel's found like potassium in the ash? How to measure that?
For potassium in the ash and all, it is like specific techniques. We can use a EDX and or spectroscopic techniques. So this is like a total mineral content. I'm not going with a particular thing.
Ma'am, in the estimation of anthocyanin calculation, there you mentioned 98.2 value, right? What is that value defined?
So that is like a factor. So it is like in the formula derivation. So we will be calculating with the factor. So that is a factor. 98.2 is a factor.
Okay.
Mm.
Ma'am, is there any other standard solution except BSA?
It is there. So Geldol will, it is different for whatever I'm taking. So the food samples is usually having the BSAs. So BSA standard is a common standard which we'll be using for our food analytical equipments. So analysis. So another one is Geldol. So for this, I'm using a food analysis. So I want to express how the Lowry's and Geldol will be there. So Geldol will be, it is like automatic. Only at the end we will be titrating. So we can use a Geldol also for Lowry's. For this workshop, I'm like giving a brief about the Lowry's. So that's what I'm choose. There is nothing like accurate than Geldol or nothing. We can estimate from the both.
But in both methods, which will give more accuracy, no?
It is depending upon your sample as well as it is depending upon your instrument calibration. If the Geldol is working good, we can go with that. Otherwise, we can also use many estimation techniques. Lowry is the most best because it is like reactions and UV absorbance, so it is best. So for preparing a sample using a solid, if it is solid, it is also fine. We can go with the dilution factor. So if you are taking 1 grams of sample, going with, if it is a powder, so 1 gram with a 10 ml of distilled water, like 1:10 ratio, like that. So after that, we have to multiply the dilution factor at the results. So that is the way we can prepare the sample for the crude protein estimation.
Ma'am, till now we have discovered like macronutrients and micronutrients, all that carbohydrate, proteins, minerals, vitamins. And what if while doing the analysis we found something new and we didn't know what that thing is? So we may discover some new means vitamin or minerals. How to know that?
For like volatile compounds, we can go with the GCMS analysis. Like it is like this is depending upon the composition analysis. So we can also go the analytical techniques, instrumental techniques like GCMS, HPLC, which we'll be discussing in the day 2, day 3 lectures. In that also we can estimate. So in EDX, the one asked question, right, for the mineral composition in the ash. So specific. So for specific estimation, we can go HPLC, Raman spectroscopy, NMR for the protein and all, and like that we can go for specific. And we will be discussing this lecture like day 2 and day 3 for HPLC and GCMS. So no worries, we will be explaining that also. Okay, then if any queries are there, we please post in the WhatsApp group. We will be discussing today itself. And don't worry about the presentations. We will be sharing the recordings at the end of the workshop. And tomorrow we will start our workshop by 10 o'clock. Please make sure that you will be available. And that's it for today's presentation and lecture. We will meet tomorrow. Thank you all.
Thank you.
Thank you, ma'am.
Thank you. Thank you.